Hello, I'm Akito Ono, Director of Cell Biology Team at Axcelead Drug Discovery Partners. Today, I'd like to introduce our phenotypic drug discovery platforms. First, let me introduce our assay platforms at Axcelead. We cover a wide range of target classes, GPCR, ion channel, enzymes, nuclear receptors, and also we have a long experience in the field of phenotypic screening. Today, I'd like to focus on phenotypic drug discovery platforms, especially compound screening process, that is phenotypic screening. Compound screening for drug discovery can be divided into mainly two categories: target-based screening and phenotypic screening. Also, phenotypic screening, assay systems mostly low throughput and low stability. Target deconvolution and analysis of mode of action of hit compounds could be challenging step because there are no versatile methods for target deconvolution. On the other hand, the good thing about phenotypic screening is we don't need to know the molecular targets of a disease when we start a screen campaign. And additionally, we have a chance to discover hit compounds with unique MOAs, which we can't find or even imagine through target-based screening. I believe this is the best thing about performing phenotypic drug discovery. Phenotypic drug discovery has been re-evaluated due to the advancement of research technologies. Novel screening tools and disease models are available, including iPS cells. There are great progresses in assay system and target deconvolution technologies. As technologies become more advanced, I think we can really succeed in phenotypic drug discovery. These are typical screening cascades for target-based screening and phenotypic screening. The difference point is after the hit compound identification process. Of course, we can move forward with lead generation and optimization stage using hit compound from the phenotypic screening, but we can also take a MOA analysis and target deconvolution approach in parallel. If we succeed in target deconvolution in early phase, we can facilitate our phenotypic drug discovery. Axcelead would like to support your phenotypic drug discovery with wide experience, state-of-the-art facilities. We cover a wide range of assay readouts under biosafety level 2 environment, including high-quantity assay, reporter-gene assay, gene expression screening, and so on. High-throughput electrophysiological screenings are also available. If you could provide assay materials, we are able to perform CRISPR-Cas and siRNA screening as well. We have experienced about 70 phenotypic screening programs. The imaging analysis is the largest categories, followed by reporter-gene assay and cell proliferation assay. As a new modality, we have a capability of protein degrader screenings using protein of interest high-pitch system from Promega Corporation. For each screening campaign, we use tens of thousands to hundreds of thousands small molecules and found a variety of hit compounds, so we can propose the best screening cascade based on your needs. As a compound library, we have a pharma-origin, high-quality small molecule libraries with a good read likeness. It consists of about 1.5 million compounds. Focused libraries are also available. When running phenotypic screening, biologically annotated compound library, for which a compound's MOA has already known, is a powerful tool to facilitate your MOA of hit compound. Since the MOA analysis of hit compound from phenotypic screening is a challenging process, we usually use Annotation library. For example, you could kinase A inhibitor as a hit compound from your phenotypic screening. Then we can search if we have other kinase A inhibitors with different chemical structures in our internal compound library. As we have experienced about 600 target-based screening, our compound library has a lot of annotated compounds. Then we check if the other kinase inhibitors induce the same phenotypic outcomes, and positive correlation was observed between cell-based activity and cell-free activity. So if yes, we could validate kinase as a responsible target molecule for the phenotype using genome editing tool. If we succeed in target deconvolution, we can start the kinase inhibitor screening project to find the modulators of your interested phenotype. This scheme could drastically accelerate your phenotypic drug discovery. This is an actual example of using Annotation library for MOA analysis. Steroid called saturase 1 inhibitor has an anti-proliferative activity, but its effect was partial. So we tried to know the underlying resistance mechanism of cancer cells against SCD1 inhibitor. We took a combination screening approach. That means we searched for compounds which are synergistic or antagonistic with SCD1 inhibitors. After investigating the annotation of possible combination candidates, we found autophagy was a key factor that modulates cancer resistance against SCD1 inhibitor. So this suggests that combinatorial therapy with a CD1 inhibitor and autophagy modulator could be one option. And also this suggested that using Annotation library for MOA analysis is a powerful tool. We are also working on compound screening using iPS cells. We are collaborating with Fujifilm Cell Dynamics Inc. and developing screening platform to provide high-quality screening service. Now we are internally working on 2 feasibility studies using iCell products, and we'll be able to provide screening service in the near future. This is one of the examples using iPS cells for compound screening. We performed compound screening for modulators of BNP expression using iCell cardiomonocytes and biologically annotated compound library. We successfully identified several hit compounds, and their pathway analysis reveals the novel engagement of each molecule in the regulation of expression of BNP expression. Now we are planning to diversify our capability into other types of iPS cells. Let me summarize my presentation. We'd like to support your phenotypic drug discovery with high-quality library, wide range of assay platform facilities, and our long experience in this field. If you have interested in phenotypic drug discovery, please feel free to contact us. We are looking forward to working with you. Thank you for your viewing.